All right, now we're going to talk about how the cell replicates the DNA. DNA is replicated in the synthesis phase of the cell cycle. Now, our DNA contains the recipes to make all of our body's proteins. And we have about 25,000 recipes or 25,000 genes. So you can think of our DNA as being like a cookbook, where each recipe is a gene. We have 25,000 of them, which would make a really huge cookbook. So we break them into volumes, and each volume is called a chromosome. Now, our cells have a 23 volume cookbook. That's how many books it takes to cover 25,000 recipes. But we also have two sets. We get one set of cookbooks from our mother and one set of cookbooks from our father. So we have a total of 46 chromosomes or 23 pairs. And all combined, this is more than 1 billion nucleotides of DNA in every single cell. More than 1 billion nucleotides that has to be copied before a cell can divide. Now, here is a picture of the 23 pairs of chromosomes. So you can see that each one of these is with its the other part of its pair. So this is chromosome number 1, chromosome number 2. This is maybe the copy from mom, and this is the copy from dad. Each one has the same genes. So we say that they are homologous. However, they may not have the exact same recipe. They may have a different variation, and the variations of a gene are called the alleles. So if these were actual recipes, and this was the recipe for oatmeal cookies, this allele or version or variation of the recipe might tell you to put raisins in. And this one might suggest peanut butter. So they're both still oatmeal cookie recipes, but they are variations of the basic recipe. This is the same thing when we have the gene to give us different color of eyes or control how our ears are attached to our head. And some of these, there are different ways of the alleles behaving. Some of them are dominant, which means that if you have that variation, that's the trait you're going to have. Some of them are recessive, which means that only if a dominant's not around is that going to show up. And there are other ways that alleles express themselves. That topic has to wait for another time. Now, during interphase, which is everything that's not mitosis, the DNA exists as what we call chromatin. It's loose and accessible so that when we need to find a recipe and make the protein that recipe encodes, then it's easy to get to. It's sort of like when you've moved into a place, you unpack your things, you take them out of boxes and suitcases so that you have access to them. When you have to move, you pack them up nice and tightly. But in between, you have things unpacked. Same thing with DNA. So it's still organized. In fact, it's wrapped around these little particles to form what we call nucleosomes. And then those nucleosomes kind of keep the DNA from getting tangled or damaged, but still make it accessible. Now, DNA is a double helix. That means that it's like a ladder, but it's twisted. The double means that there are two strands. Each half of the ladder is a separate strand. So here's one half of the ladder, here's the other half of the ladder. And the ladder is held together in this gap by hydrogen bonds. Individual hydrogen bonds are very weak, but when you have a whole bunch of hydrogen bonds, it sort of acts like a zipper that keeps these two strands together. Now, how did we figure that out, that crazy shape of DNA? Well, Dr. Rosalind Franklin used a technique called X-ray crystallography to determine the structure. When we talked about isotopes earlier in the semester, this is one of the things that I mentioned that isotopes are useful for. So she used X-rays and shined them through a DNA crystal and got this pattern, and from that somehow, because I can't tell, figured out that meant that there was a double helix. Now, Dr. Franklin received the Nobel Prize for her work in 1962. Just kidding. The Nobel Prize was actually awarded to these two guys, Watson and Crick, who actually stole her data and took credit for her work. She died in 1958 at the age of 38 from ovarian cancer, quite possibly due to the exposure to radiation in her lab. She has never been awarded the Nobel Prize for her work because they don't give it to you after you're dead. Not really fair. Okay, let's get back to the structure of DNA. Remember that nucleotides are made of three basic components. Every nucleotide has a sugar that has a phosphate group attached to it and a nitrogenous base. Now, the nitrogenous bases come in different forms, but all of them have lots of nitrogen. In DNA, the sugar is called deoxyribose, or ribose with an oxygen removed. So DNA actually stands for deoxyribonucleic acid. Now, nucleotides are joined together to build a nucleic acid through dehydration synthesis, the same process that every organic molecule does in our body. So this forms a bond between the phosphate group of one nucleotide and the sugar group of another nucleotide. So that what we have in the end is a long chain that looks like this: phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on, where they're all attached to each other, and then the nitrogenous bases are exposed. So we call this the phosphate sugar backbone. It forms the vertical support of the ladder. And the rungs of the ladder, or half of the ladder, are the nitrogenous bases. Okay, so the DNA is two of these strands facing each other, making the ladder shape. So this is our phosphate sugar backbone. These are the nitrogenous bases, and hydrogen bonds are holding everything together. If we look over here, same thing. This is the phosphate sugar backbone. These are the nitrogenous bases, and those dotted lines are the hydrogen bonds that are holding them together. Now let's talk a little bit about the nitrogenous bases. So DNA uses 4 of them. They are adenine, which when we put it on the sugar and phosphate, it becomes adenosine. It's the only one that has a name that changes. Thymine, cytosine, and guanine. You'll notice that some of these have single rings, some have double rings, but no matter what, we see lots of nitrogen. And as it turns out, these guys like to bond together, but in a very specific way. So, adenosine or adenine always has hydrogen bonds with thymine. I'm trying to get this so it doesn't cover everything. All right, so these two always go together. Adenosine does not make a connection with cytosine or with guanine. And cytosine and guanine always form hydrogen bonds together. So there are lots of ways to remember how this goes. I always remember that the straight letters, the ones that are made with straight lines, go together, and the round letters go together, the C and G. So you may find other ways to recall, but that's always been the easiest for me. And the fact that they like only certain complementary nucleotides or nitrogenous bases, it gives us something called complementary base pairing. So what that means is that if I have an A on this strand, then I know there's going to be a T on this strand. If I have a G on this strand, then I know that on the opposite strand, there's going to be a C or cytosine. And this is the key to the cell being able to copy its DNA. Okay, now, in order to copy the DNA, first we have to unzip the DNA so that we can see the nitrogenous bases inside. So there's an enzyme called DNA helicase. Helicase, ace means enzyme, heli means the helix. So this is taking apart the helix or unzipping the helix. And it slides along one of the strands and unzips the DNA as it goes. After the helicase has exposed the strands, the enzyme DNA polymerase, All right, so this actually tells you what it does. ACE is enzyme. Polymer is multiple monomers putting together, and then DNA. So this is an enzyme that makes polymers of DNA. Now, it only uses DNA nucleotides. That means that this enzyme can tell what sugar is that nucleotide is made with. And if it's made with ribose, which is RNA, it doesn't want it. But if it's made with deoxyribose, it'll take it. And whatever is exposed on the original strand, it will look for the complementary nucleotide and put them together. Now, this means that our resulting DNA strand is what we call semi-conservative. So the blue is the original strand. So you can see that there's one blue strand. The yellow is the new strand that the DNA polymerase is building. Now, the DNA polymerase puts the nucleotides together using dehydration synthesis, which we saw before. And then we have a new strand of DNA. This is done on both sides, of course. So we end up with two semi-conservative copies of DNA, and now the cell is ready to divide.